So we're going to conclude the course today looking at the effects of plant chemistry on plant growth and development, and also in defense of plants. This is a little bit sketchier than some of the material we talked about up to this point with regard to anatomy and morphology. These aspects of plant physiology are still being studied actively. A lot of progress being made with molecular biology approaches being applied to plant physiology regarding growth regulation. But there's also a lot known, and we're going to go over some of the brief, just give you sort of a brief overview of some of this information, which is really pretty different from what goes on in animals as far as growth regulation. So last time ended by talking about plant photoreceptors, how plants actually perceive light, and we're going to go from there then into looking at how a signal like that that's received is translated into a response on the part of the plant involving plant hormones or growth regulators. So last time we ended on photoreception, talking about photoperiodism, which is a really important topic for many plants that have photoperiodic responsive flowering. So many plants are either short day plants or long day plants that only flower once day length reaches a critical threshold. And it's actually not day length, as I mentioned last time, but it's night length that the plants actually appear to be measuring. And there are a lot of experiments to demonstrate this that basically tried all different kinds of manipulations of light and darkness to see what really affected the signal or the triggering of plant flowering. And it turned out it was uninterrupted night length, not day length. That was the key thing, as I mentioned at the end of the last lecture. And what was found was that in short day plants, for example, that's those that are flowering as the night length crosses a critical threshold here, as you can see. If you actually impose a brief but bright flash of light in the middle of a dark period prior to exposure of these plants to a period of night that's sufficient to trigger flowering, here's that flash of light, the plant will not flower. And what's happening here is the plant's basically measuring the uninterrupted night length, and neither of these intervals on either side of this flash of light is sufficiently long to cross this critical threshold of length needed to initiate flowering. And in long day plants, where we have to have night length basically decreasing beyond a critical threshold in order for flowering to occur, if you impose a flash of bright light in the middle of that dark period, it was too long for flowering to be initiated. The plant now sees an uninterrupted night length that's below that critical length threshold here, and flowering is initiated. And this is pretty well understood in terms of the kinds of photoreceptors that we talked about last time. This involves phytochrome. The photoreceptors are in the red, far red, into the light spectrum. And it's the ratio of phytochrome red, one form of this molecule, relative to phytochrome far red, another form of the same molecule, they're interconvertible, that dictates, or that basically allows plants to measure that night length. And so the form phytochrome far red gradually is converted to phytochrome red under darkness. And that's the clock, basically, that plants use to detect night length. But even a brief flash of light is sufficient to convert phytochrome red to phytochrome far red. And so this is the way in which this whole mechanism works, is based on that conversion of red to phytochrome far red between those two forms. So that induction response is understood to involve some sort of transmission of a chemical through the plant. And one experiment that beautifully shows this is that you can take a long day plant like this one on the right here that has never been exposed to sufficiently short nights to initiate flowering. You can see the night length here it's been grown under is too long for flowering to occur. And you can graft this plant onto a plant here on the left that's been exposed. It's a short day plant that's been exposed to the same night length, but in the case of a short day plant, that's past the critical night length threshold to induce flowering, and flowering's already begun. You graft this long day plant onto this short day plant, and flowering is initiated in the short day plant. And it's never been exposed, this short day plant's never been exposed to sufficiently, or sorry, this long day plant here grafted on has never been exposed to sufficiently short nights to initiate flowering. So this suggests that there's some chemical transmission between these, between the plant that was induced and the graft here that results in the flowering response. And plant physiologists have struggled for over about a century to find out what that chemical is. And it's been sort of the holy grail of plant physiology to find the elusive plant hormone, flowering hormone called florigen. So it's had a name for as long as anyone can remember, but nobody actually knows what it is. And there are some suspicions now that they've been looking in the wrong place for it, that it might be a macromolecule rather than a micromolecule, something like a mRNA or a protein that's actually involved in this. And there's some suspicion that it's a protein produced in the leaves that's transmitted through the phloem to the shoot apex and triggers the change from a vegetative shoot apex to a reproductive shoot apex. But it's still being studied, and it's one of these lingering mysteries of plant physiology, which we'll get into more in a minute. But this all fits within sort of the general area of plant chemistry, which is incredibly diverse. Plants produce an incredibly diverse array of chemicals, a lot made them important in medicine. And part of the reason we think that plants are so rich in chemistry has to do with their inability to move once they're mature sporophytes. In the case of vascular plants, they can't just get up and move away from negative stimuli or move toward positive stimuli like an animal. They have to basically just deal with it. And the way that the plants deal with environmental change is either through some sort of metabolic change, in the case of something like this resurrection plant, which is a lycophyte that is one of the few vascular plants that has desiccation tolerance, so it can just go into a dormant desiccated state when there's a drought. Or plants can initiate some kind of growth and developmental response. And that's commonly what plants do, is that they undergo some sort of growth response that deals with a major environmental change, like becoming shaded, experiencing drought, those kinds of things, lack of water availability. All right, so the compounds that mediate these growth responses, these growth and developmental responses, are commonly called plant hormones. And this terminology has been in place for over a century. That is to call plant growth regulators hormones. The term hormone was first coined for animal growth regulators. Of course, we have an endocrine system, and it's much more straightforward in animals by comparison with plants in terms of studying these growth regulators, because plants don't have an endocrine system. And their hormone action, their growth regulator action, is extremely complex and relatively difficult to study. And some people question whether this term hormone really should be applied to plant growth regulators. But they do show a lot of similarities to animal hormones in some important respects. For instance, they are organic compounds that modify or control one or more physiological processes within the plant. They're also transported from the place where some sort of stimulus is received, for example, by a photoreceptor, to a place where the action of the hormone is needed, the point where a growth response or developmental response is needed to respond to that stimulus in an effective way. Sometimes, though, that distance of transport is extremely minimal, and in that regard, it's different than animal hormones. But like animal hormones, plant hormones, as they're called, are often effective in regulating growth at really low concentrations. So these things operate to result in a response at extremely low concentrations, and they're involved in just about every part of plant growth and development. The really thing and part that makes hormones really difficult to study in plants is that an individual hormone can be involved in many different kinds of responses, many different kinds of growth or developmental responses. This can have to do with the site of action. So one organ in the plant, like the root, might respond to a hormone really differently than the stem does. So it depends on where the hormone is acting, and the concentration of the hormone can matter a lot in terms of what that activity is. And that can vary between organs and between tissues, as we'll see. And it also can matter what developmental stage the plant is at, where it is in its life trajectory can influence what the impact of a hormone is. But another thing that makes things complicated with hormones is that an individual growth response, a particular response that we can detect and measure in plants, might be triggered by multiple hormones at once. And it's really the relative concentration of these hormones that's important in eliciting a particular response in the plant. And so this term hormone balance, which no doubt I've heard about with regard to human physiology, is especially important in plant physiology. All right, so to introduce the action of hormones, we're going to start with the first plant hormone that was actually detected. And this was detected in the research of none other than Charles Darwin, who's someone who we often look to for early insights in biology and evolution. And he did some interesting experiments late in his life with his son Francis that were really key in plant physiology. And he was looking at what causes plants to grow toward light. So if you put a plant on the windowsill and leave it there, you'll often notice that it'll bend toward the light, and it'll grow in that direction. And if you turn it around away from the light, it'll bend, it'll grow back over toward the light again. And so that was long known, but this was something that wasn't understood. So they decided to study this by growing grass seedlings. And remember, grasses like corn, we showed monocot germination earlier. The coleoptile, the sheath that surrounds the shoot of a developing monocot like a grass, emerges first from the soil, grows straight up, and then later the shoot emerges out of that sheath. The sheath protects the shoot as germination occurs, and it's these coleoptiles that Darwin and his son were studying. And so they grew them. The control was grown with light occurring to one side of the plant, and the coleoptiles bent toward the light as expected. But if they cut off the tip of the coleoptile prior to this light being directed toward the coleoptile, there was no bending response at all. And if they didn't cut off the tip, they just covered it up with an opaque cap so that light couldn't reach it. We also didn't get bending, so it wasn't just the result of some kind of wounding here that didn't result in the bending. The lack of light being able to reach that tip was sufficient to prevent bending. And also, if they covered up the part of the coleoptile where it looked like the bending was actually occurring, the bending actually still occurred. So the conclusion from their experiment was that light is sensed at the tip of this shoot, at the tip of this coleoptile, and that some sort of signal was transmitted down the coleoptile to the responding tissue that resulted in this growth response that resulted in bending. So that was Darwin's hypothesis, this scenario here. And that's as far as they took it. But this became a source of a lot of interest. A few decades later, a Danish biologist, botanist, showed that this phototropic signal that moves down the coleoptile was actually a chemical. And the way that was demonstrated was by cutting off the tip of the coleoptile and before it was exposed to light, and then putting a permeable little block of agar between the tip of the coleoptile and the base of it. And they still saw the bending response here. So this is a permeable block here, as opposed to when if they cut off the tip and they put an impermeable barrier in between the coleoptile tip and the coleoptile base, there was no bending response. And so the conclusion here was that some sort of chemical had diffused through this block, was prevented from diffusing through an impermeable barrier here, and it was that chemical that was responsible for that bending response. All right, so that's where things stood until the 1920s, and then the timing's not important, but there was a botanist of much acclaim, Fritz Wendt, at Caltech, that basically figured out that this hormonal response can occur in darkness. And the way that this was shown was by cutting off the tip of the coleoptile that had already been exposed to light, putting it on an agar block again, letting that chemical diffuse into the block. It was already clear that that would work based on the previous experiment. And then the coleoptiles that had been in darkness, but had their tips cut off in darkness, this agar block that had been allowed to be permeated with whatever chemical was induced here in the light, if you put it over the whole tip, the thing would just grow straight up, but it would elongate. If you put it off to one side, like shown in these two cartoons here, the coleoptile would bend away from the side on which the block was placed. And this all happened in the darkness. And so the idea here was that something is happening. This chemical that's transmitted down from the block is causing elongation on the side on which the block is placed. So there's some kind of the chemical is actually resulting in some sort of growth response on this side of the coleoptile. When we looked at it anatomically, it was seen that the cells on that side that had undergone the bending, or where we have this elongation, what's happened here is we haven't had cell division, but just cell elongation on that side. And it was found that the chemical that was in this block that was responsible for this, a colleague of Wendt figured out it was indolecetic acid, which is what we call auxin. And auxin is the term that was used before it was known what it was. Auxin just is Greek for to grow, and so it's just indicating it's a growth hormone. And this was the first class of hormones that was characterized in plants. Well, it turns out, unfortunately, that coleoptiles are actually different than shoots in their growth responses, and this doesn't explain how the shoots of plants actually bend toward light. It's specific to coleoptiles, but it was subsequently seen that auxin has a number of really important impacts on plant growth and development in general. And we're going to look at some of those examples now. So that's just how it was discovered. But auxin turns out to be produced in the shoot tip, so in the terminal bud or apical bud of a plant that's undergoing active growth, producing new stem and leaf tissue. It seems to be produced there and transported down the shoot. and it's actually transported in the xylem. But in the parenchyma cells, I mean, obviously the things that are moving within the trachees and vessel elements are moving up through the plant. There are living cells in the xylem, parenchyma cells, and this is the auxin signal is transported down through those cells pretty quickly relative to what would happen through mere diffusion. And so remember, there are these axillary buds in the upper angles of plant leaves from which shoots can develop, from which branches can develop in plants, and they were set up during the early stage of development of the shoot, from the bud primordia. Well, those axillary buds, we often see that branching occurs generally more readily lower on the plant, that is further away from the shoot apex than it does close to that shoot apex. And just think about the normal shape of a tree. You have larger branches near the base and shorter branches near the tip. Branching is initiated. Anyways, the general pattern we see is that branching occurs after the shoot apex basically gets a certain distance away from those axillary buds. And it turns out if you cut off the axillary bud, if you cut off the shoot apex, you will see, like you see here, although this is time-lapse, it's not quite this fast, obviously, but you see that these axillary buds will be initiated and start rapidly growing, and usually straight up like this in the case of this kind of wound situation, and one of those will end up becoming the dominant shoot apex at that point. So the general hypothesis has been that auxins produced at the shoot tip, transported down the stem, down the shoot, and it inhibits the buds. And if that inhibition is removed, for example, by cutting off the tip, then you'll get a lot of branching occurring. There have been some challenges to this idea fairly recently based on some recent experimental results, but this model is still, a lot of observations are consistent with this hypothesis. And we're going to come back to this, so hold on to this thought, because we're going to come back to this in a moment with regard to hormone balance. It's a really pretty interesting case of hormone balance that really dictates how the shoot grows. All right, so auxin also has a major role to play in the growth and development of fruits. And auxin is produced in the vicinity of seeds after seed set occurs. And if pollination doesn't occur, and we don't have fertilization of the seeds for whatever reason, generally, oftentimes at least, the fruit won't develop normally. And that's actually a good thing for the plant, not to waste effort on developing a fruit if it's not going to set any seed. It's just a big waste. But if you actually spray the ovary, for example, of a tomato with auxin, even if there hasn't been pollination and there is no seed set, the fruit's going to develop normally except for the lack of seed set. So auxin definitely has a role to play in the growth and development of fruit. So if we look at auxin's effects, it only actually, in terms of its growth stimulating effects on stems and coleoptiles, it's involved in cell elongation, but not in division or differentiation of cells. And the way in which auxin is suspected to operate, and it's been pretty well studied, is that it triggers these enzymes called expansins that basically modify the cell wall of cells, basically breaking microfibrils in the cell walls, allowing the cell to expand. It actually also affects the membranes of cells such that will have the osmotic properties of the cell change such that the cell will actually expand and become there'll be some positive pressure that promotes that expansion in the face of the weakening of those cell walls. So there is a lot known about auxin's action. Also, as I mentioned, apical dominance of the shoot, and we'll get to that again in a moment, as well as stimulating fruit development. And also, as we'll get to in a moment, even though it inhibits branching of the shoot, it promotes branching of the roots, the formation of lateral roots. So it has the opposite effect in the roots. So if we look at different parts of the plant, you can see that there's different levels of sensitivity to different concentrations of auxin. And here you can see the concentration of auxin from low to high on the x-axis. And here we can see the growth effect on the y-axis, positive from here up, negative from here down, increasingly negative, increasingly positive. And so you can see for roots, axillary buds, and stems, for any one of these organs, we have a different type of response at different concentrations of auxin. So there are some levels of auxin that have a strong positive response on root growth, and some that have higher concentrations have a negative impact on root growth. And the same is true for buds and stems, and these different plant parts differ in the levels of auxin that initiate a certain type of response or lack of response. So, for example, in stems, the maximum growth response that we can get as far as elongation of stems is actually the most inhibiting response to the axillary buds. I mean, at that level, at auxin concentration, you get strong inhibition of the axillary buds. And similarly, at the level of auxin that's correlated with maximum growth of the buds, we get maximum inhibition of the roots. So you can see how these concentration effects can really make a big difference depending on, well, not just on the level of concentration for a given plant part, but depending on which plant part you're looking at. All right, so that's all pretty oversimplified. It's pretty simplified because it's really the hormone balance that plays the biggest role in all of this. These hormones are not operating by themselves usually, and nowhere is that truer than in the case of shoot and root growth. As I mentioned, the auxin's only operating to promote cell elongation. It operates in concert with cytokinins in overall growth response, and cytokinins promote cell division and differentiation, the other aspects of growth in addition to cell elongation. And it's the cytokinin to auxin ratio that's going to dictate the growth outcome for different parts of the plant. So a high cytokinin to auxin ratio, that is, the cytokinin's high relative to auxin, that's going to promote activation of shoot branching. That is the activation of those axillary buds.